pharmacogenomics is an emerging field in the area of personalized medicine. So personalized medicine means a particular medicine will affect or will give a positive response toward a particular person having a particular genotype. So by saying genotype, okay, so we will try to understand the genotype, right? So every individual has is specific genotype, although entire genome is 99.9% matching with all the genome, right? All the person genome, they are matching, but there is a slight variation in a single nucleotide that differs from one person to another, and that makes the response towards its environmental condition, their environment. So here environment I am saying drug in the case of pharmacogenomics. So let us understand this. So every individual has a particular genetic makeup, okay? So that that particular makeup will respond towards a particular drug in a particular fashion. Some person will respond towards particular drug in a better way. Some will respond in a medium way. The other will not respond at all towards a particular drug. Okay, so this is a diagrammatic representation where we are showing like a population of population having no response towards any drug, a desired or a good responder, or a no is a the other one is the serious or side effect or medium effect of that particular drug. So I think up to this point we are understand.
Yes, sir.
Okay,
Yes.
so if I will ask you, so what is pharmacogenomics? Can anyone explain right here?
Yes,
Pharmacology
sir.
and
Definitely.
genomics are combined as a pharmacogenomics.
Okay, so this tells about the response of a particular individual towards a particular drug is the study of pharmacogenomics, okay, or the pharmacogenomics is the study of a particular drug, okay, of a particular drug of a particular individual. So a particular individual will respond towards a particular drug in a particular way depending upon its genomic constituents or a genomic makeup, we can say. Okay, so moving towards the other slide. So actually this pharmacogenomics no, due to the implementation of this area in patient care, so it improves actually the patient care, because we are giving drugs to a particular person in a particular way, okay, in a particular dosage, right? So every individual has their different BMI, okay, so weight, height, everything. So according to that also we are recommending whether this much of drug has to be recommended for this particular patient, okay, so that improves the patient care. So customized treatment plan, that means this particular term you will understand in the succeeding slides, how we are saying like we are customizing the treatment for a particular patient, and also we are minimizing the adverse effect of particular drug. Of course that drug will have a positive response or give a positive response to a particular patient, but if we increase the dose of a particular patient, that will have an adverse effect on that, right? So that has to be tailored, or that has to be customized, and we are customizing that particular drug in this pharmacogenomic test, right? Also for a particular disease there is a very kind of drugs are available, Okay, so this test helps to filter all those not required drugs, and we are giving a particular drug in a particular concentration or a particular dosage, so that they will give a specific response, or patient will have benefit from that drug, right? Previously what was happening is like, no, if a particular patient does not no response to a particular drug, so hit and trial and everything, the doctor has to do hit and trial to stabilize on a particular drug, like if this drug is not working, we are giving this drug. But not in pharmacogenomics. Pharmacogenomics, where we study the genomic part, okay, their genetic makeup, and according to their genetic makeup, we are suggesting the drug and its concentration or its no amount, how much they should take, and that amount will give a positive response, right? So the other point is like reduced treatment failure. Of course, if you'll give drug on a basis of their genetic makeup, that will of course reduce their no this treatment failure. Okay. The other point is prescribed with more confidence. So since we are giving drug on the basis of its genetic makeup, we are 100% confident that this particular drug will work, and this particular dosage will work on that particular patient. So up to this point, everyone is clear?
Yes, sir.
No, no. I need to know some question from this slide. If no one needs, no. I think if anyone is having any question on this slide, any question, any question. Okay, fine.
Sir, good morning. I just wanted to know, like, this will work with immunodepressant drugs also or not, sir?
So we will discuss that. We will discuss this part. Of course, we are working on the immunodepressant drugs. So we'll discuss in detail that particular drug. We'll discuss in detail. Okay. In the next slide, we will be discussing all those stuffs. So I think this slide is clear to everyone. So first slide is clear to everyone, why we are giving a particular drug to a particular patient, and that is studied under pharmacogenomics, and what are the precautions, and what is the improvement of pharmacogenomics over other treatment. So this particular slide tells about that. Now, as asked by someone, I don't know the name, so we'll move towards the other slide. Now, these are the four pharmacogenomics study or test which we are targeting at Sandor. So tacrolimus pharmacogenomics, warfarin pharmacogenomics, clopidogrel pharmacogenomics, and anti-epileptic drugs pharmacogenomics, right? So these are the four target area where we are more concentrated towards in pharmacogenomics. So you can just see the slides, like tacrolimus pharmacogenomics is like analysis of FDA approved genetic variants of MDR1, CYP3A4, and CYP3A5. So these are the genetic variants. Okay, so MDR1 is a gene, and CYP3A and CYP3A5 is of course a gene. These are the genes. So these are the genes where particular variants are present. Okay, these particular variants differ in a person. Okay, so somewhere A will be there. Just giving an example, so in some person at a particular position, A will be there. At that particular position, some person will have a G, okay, and some person will have a T. This is an example I'm giving you that I will discuss in detail in the other slides. So if A is a good responder towards a particular medicine, okay, and G is an intermediate, and T, for example, T is an intermediate, okay. So according to that particular genetic makeup, we are approving or we are suggesting doctors like give a high dose, give intermediate dose, or change the drug, right? Similarly, warfarin pharmacogenomics also actually works upon three different no, variants like CYP2C9, VKRC1, and GGCX. So these are the different genes we are targeting, and in that particular this three particular gene we are targeting uh, different variants in that. According to that variant, no high responder, medium responder, and no responder or low responder. So accordingly, we are suggesting drug. Similarly, for clopidogrel and anti-epileptic drugs also. So up to this point, any confusion? Please
Yeah,
ask
we are
Sir,
question.
good.
could you please repeat this slide, please?
This entire slide.
Yes, sir.
Okay. So at Sandor, we are targeting this. This slide is visible to everyone, right?
Yes, we are visible.
Okay, this is visible. So at Sandor, we are targeting under pharmacogenomics department. We are targeting this four pharmacogenomic test. The first one is tacrolimus. The second one is warfarin. The third one is clopid, and the other one, the fourth one is anti-epileptic drugs. Okay, so first I will go one by one. So
Yeah.
tacrolimus. So
Dr.
tacrolimus is an
Santosh, एक मिनट. Dr. Santosh, 1 minute. हाँ, Syed here, okay? So, guys, actually, Dr. Santosh जो बता रहे हैं, these 4 drugs are very long-term use drugs. For severe diseases like tacrolimus is for nephrology where kidneys are damaged. तो kidneys के लिए warfarin and tacrolimus can be given there. Clopidogrel for cardiac patients जो long term जिसको स्टंट डालते हैं या तो फिर किसी का बाईपास सर्जरी होता है तो उनके लिए क्लोपिडोग्रेल ड्रग है जो लॉन्ग टर्म मतलब जब तक वो रहेंगे बहुत लॉन्ग यूज करना पड़ेगा तो इसके लिए जब लॉन्ग यूज करते हो तो ड्रग पता नहीं चलता है ना कि काम कर रहा है एक बार यूज करते 6 महीने साल भर इसके बाद में आई थिंक दैट विल बी लाइक रेसिस्टेंस या तो ड्रग डोज बराबर बस आई मीन काफी नहीं हो रहा होगा उसकी ब्लड की विस्कोसिटी क्लोपिडोग्रेल की शायद उसका इफेक्ट बॉडी को नहीं हो रहा तो फार्माकोजेनोमिक्स इज ए टेस्ट we will say that whether this drug is suitable और वर्किंग और एफिकेसी उसकी है या नहीं है टू आइडेंटिफाई एंड टेल द डॉक्टर कि भैया ये ड्रग इनको काम कर रहा है या फिर इनका डोज चेंज करना पड़ेगा या तो फिर कम करना पड़ेगा तो डॉक्टर डिसीजन लेता है इस ड्रग के बेसिस पे टेस्ट के बेसिस पे फार्माकोजेनोमिक्स के टेस्ट के बेसिस पे तो सेम एपिलेप्टिक एपिलेप्टिक भी जो एपिलेप्टिक का ड्रग है वो ड्रग्स भी आपको लॉन्ग टर्म यूज करते हैं जिसको बोलते हैं ना फीट्स आता है सीजर्स होते हैं सडनली लड़के लोग गिर जाते हैं तो दैट हैज टू बी यूज्ड फॉर लॉन्ग टर्म वेर एवर देर इज ए लॉन्ग टर्म यूसेज ऑफ ड्रग्स जितना लॉन्ग यूज करना होता है तो पता नहीं चलता ना कि ड्रग काम कर रहा है कि नहीं कर रहा है मे बी 6 महीने बाद यूज करने से फिर भी कुछ पेशेंट्स डॉक्टर के पास अगर आप लोग अपनी फैमिली मेंबर्स में अगर किसी को ऑब्जर्व किए हो कि यार ये ड्रग शायद ये डोज तो ले रहे हैं बट स्टिल हमको प्रॉब्लम हो रही है या तो फिर हमको कोई कोई आके कंप्लेन करता है डॉक्टर की यार डॉक्टर साहब हम लोग ये ड्रग ले रहे हैं मगर हमको चक्कर आ रही है या तो वॉमिटिंग हो रही है तो मतलब इसका डोज ज्यादा हो रहा है या कम हो रहा है ये ड्रग कि जितनी एमजी या जितनी भी लिमिटेड ड्रग अवेलेबल होती है दवाई में दैट इज sufficient or not for the patient to identify this test will be helpful so Sandor is doing only 4 drugs as of now वो 4 ड्रग है टैक्रोलिमस एक ड्रग है वारफरिन एक ड्रग है और आपका एंटी एपिलेप्टिक एंड वन मोर इज
clopidogrel.
क्लोपिडोग्रेल सो गाइस दिस इज व्हाट फार्माकोजेनोमिक्स सिंपल पॉइंट्स में बाकी टर्मिनोलॉजी डॉक्टर संतोष समझा देंगे ठीक है और आई वांट ऑल द गाइस टू गेट इन्वॉल्व इन दिस ट्रेनिंग गाइस अगर आप लोग नहीं पूछोगे संतोष जी एक बार लैब में घुस जाएंगे फिर यार बहुत इतना टाइम नहीं दे पाएंगे शायद दे पाए मगर इतना टाइम नहीं दे पाएंगे क्योंकि वो अपने लैब ऑपरेशंस में बिजी रहते हैं सो प्लीज गाइस आस्क दिस इज द फाइनल ट्रेनिंग फाइनल डे ट्रेनिंग सो आई वांट एवरीवन टू गेट और ये प्रोडक्ट बहुत चलता है और कोई ज्यादा कंपनीज करती नहीं है दिस इज व्हाट द एडवांटेज देखो अगर एक्सोम न्यूबॉर्न स्क्रीनिंग में हमको बहुत कंपटीशन मिल सकता है फार्माकोजेनोमिक्स में बहुत कम कंपनी से एक या दो कंपनी से सो गाइस मेक श्योर दैट यू लर्न दिस एंड मैं इसके बाद में कमर्शियल ट्रेनिंग में बता दूंगा कि टू हुम वी हैव टू टारगेट दिस प्रोडक्ट्स ओके
Yeah, okay. So
Sir,
thank you very much
sir,
explaining all
sir,
those.
Munawar here.
Yeah, Munawar ji, good morning.
सर ये टैक्रोलिमस के साथ सिरोलिमस भी होता है ना सर?
Sorry, synonymous?
सिरोलिमस
Yeah.
होता है, हम
ये
लोग
भी
कर नहीं रहे।
फार्माकोजेनोमिक्स में ही आता है
मुनव्वर,
क्या?
Yes, yes,
मुनव्वर,
yes.
हम लोग अभी नहीं कर रहे। हम वी आर सैंडोर वर्किंग ऑन लॉट ऑफ ड्रग्स टू कमिंग इन फार्माकोजेनोमिक्स। एज ऑफ नाउ सिर्फ 4 ड्रग्स कर रहे हैं।
Correct.
4, ओके। We are going to
क्या
come with
ओवर
lot of other drugs, but as of now
सर
there are only 2 drugs. Okay,
No,
सर
sorry,
since
4
सर एक
drugs.
actually this
क्वेश्चन।
Four
everolimus and
drugs.
sirolimus are in the same class, that's why they are asking. This everolimus and sirolimus is in the same class. No, we are doing
यस
pharmacogenomics
सर।
in.
Yeah, actually,
और
no.
सर
For those we don't have no. Right now we have not studied no their genetic variants right now. So tacrolimus right now we are targeting. Okay, so
सर
targeting these 3 variants for tacrolimus. We are doing it right now. So we'll of course we'll expand our wings on those areas also.
सर वन मोर क्वेश्चन। सर ये हम टैक्रोलिमस में और क्लोपिडोग्रिल में कितने जीन्स कर रहे हैं, कितने
Yeah,
वेरिएंट्स कर रहे हैं? क्योंकि
4
यहाँ पर
4
एक
genes आते हैं, CYP, A4, A5, MDR1, 3 या 4 जीन्स आते हैं। आई थिंक मोस्टली ये 3-4 जीन्स इन्वॉल्व होते हैं टैक्रोलिमस में, तो वो 3 जीन कर रहे हैं। ठीक है, 3 और 4 जीन्स आई थिंक।
tacrolimus 4 कर रहे
CYP,
हैं.
A3, A4, A5, MDR1,
Yeah, 4.
4 जीन्स हो जाते हैं। ठीक है,
टैक्रोलिमस में 4 हो जाते हैं। सो क्लोपी में 3 हो जाते हैं, वारफेरिन में 3 है और एंटी एपिलेप्टिक में 2 है।
अच्छा सर एक ओवरव्यू में बता सकते हैं, और ये फार्माकोजेनोमिक्स में और किस किस डिपार्टमेंट्स के भी टेस्ट होते हैं? ओके हम तो यही कर रहे हैं, लेकिन जस्ट ओवरव्यू
मुनव्वर,
और कौन कौन से?
मुनव्वर, अभी अभी उसमें नहीं जाते हैं। तुम्हारे को आइडिया है, यू नो ऑल दिस सर्विसेज वेल, आई नो, बट पॉइंट इज कि as of now we are doing 4 and we will speak on only 4 drugs. Let us get into all the basket of pharmacogenomics products. We will personal jab training will call everyone to Hyderabad for physical training. That time when we launch we will surely explain you all these things, I think.
या मुनव्वर जी,
ये हो
we will
जाएगा। बहुत
talk
सारे
all those
बच्चे
no
नए
stuff.
हैं। I just don't want to get ठीक है।
Munavar ji, we will discuss all those no other areas in pharmacogenomics
ओके
on
सर,
call. Okay, right now because
ओके
it's an task today. So there are
ओके सर
new candidates and new joinees are there. So they have to
सर
understand this first, right? So
ठीक
if
है
I
सर।
will give you an entire projection of pharmacogenomics, so that will be
सर
very difficult for them to understand. Right now, since I have targeted these 4 classes of pharmacogenomic test, in the
सर
succeeding slide, we will be discussing only tacrolimus no pharmacogenomics to let them understand one test first. Okay, this is an overview of all the tests which we are doing. In the succeeding slides, we will be understanding only one test. Okay, so
ओके सर।
Sayyed sir, thank you very much for explaining all those. So someone has asked me to no repeat this slide. So I was in between. So tacrolimus is no, we are giving drugs on the basis of FDA approved genetic variants, right? Warfarin also we are giving FDA approved, clopi also, and anti-epileptic drugs also. So tacrolimus consists of 4 different variants. Okay, warfarin consists of 3 different variants. Clopi also 3, and anti-epileptic 2 variants we are targeting right now. So let us understand this tacrolimus. Why tacrolimus? Why clopi? Okay, so tacrolimus is an immunosuppressant. Okay, so immunosuppressant uh, is a drug or is a compound that suppresses the no immune cells from reacting with the other alien molecules. The alien molecules can be a cells, can be drugs, or other things also, or other chemicals also. Okay, so when we actually that is particularly this tacrolimus is particularly suggested for no patient they are going undergoing this liver, lung, or heart transplant, right? So since we are transferring someone's heart or lung, okay, or some liver from one patient to the other patient, So for that particular patient, that particular organ will act like an alien thing, right? So our immune system won't be able to recognize that particular organ on the basis of immune cells. So immune cells will start invading and will try to reject all those the transplanted organs, right? So to suppress this, tacrolimus is a compound, or you can say a drug, that suppresses the activities of those immune cells. Okay, and that in that case, the patient have a successful transplant. Okay, understand? No? Hello. Yes. Mm-hmm. 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 Yeah, actually, no. Uh, this rejection is basically based upon the MHCs. MHC, I will explain. I don't want to explain, but since you have asked it, so MHCs are no the major histocompatibility complexes, right? That they are the origin of immune cells. Okay, and that immune cells is specific to a particular patient, not patient, particular individual. Okay, so since if you are transferring something from some person to another person, so that particular organ become alien, and immune cell will try to invade that. This is the thing I think you have asked, right? Yes, 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 yes. Correct, correct, correct. Since see, so actually typing you are saying, right? So actually is a you can say is a sub part of MHC. Okay, MHC is the major part called major histocompatibility complexes, right? These are the no part present on the no chromosomes, right? So they decides the type of immune cells. Okay, one particular individual have a different immune cells, the other individual will have a different immune cells. So if I will try to mix all those things, so they will try to know fight between, there will be a fight, there will be a war between these two immune cells, because they will understand like they are the invaders. Okay, so HLA typing is of course done, okay, just to determine whether I mean they are similar. We can transfer this particular organ from this particular person to this particular person. So this is a screening kind of thing, but this test determines the drug level. Okay, so we'll go to the other test, clopidogrel. So clopidogrel actually is an no anti-platelet medicine. Actually, it is. So what does it mean? Some person due to drug, they start, they in the blood there is a cell called platelets. Okay, so they are actually the major cause of, or they are the only source where coagulation of blood start at a particular location in the arteries, right? So to avoid this blood coagulation, so this particular clopidogrel drugs are given. This particular drug is recommended for those person having 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 यू कैन से जिसको हार्ट अटैक को का प्रॉब्लम हो सकता है, ठीक है ना, स्ट्रोक का प्रॉब्लम हो सकता है, सो दिस पर्टिकुलर ड्रग इज रिकमेंडेड फॉर देम, वेयर दिस हार्ट अटैक्स एंड ऑल दोस स्टफ कम फ्रॉम the coagulation of blood in the arteries. So this is an anticoagulant for these platelets. So platelet won't accumulate at a particular arteries, and this heart attack and other similar problem won't arise. Okay, similarly warfarines and anti-epileptic drugs also. So these are the 4 tests which are we are targeting right now. So in the next slide we will try to understand one drug, that is tacrolimus. Okay, so as I explained in the previous slide, like tacrolimus is an immunosuppressant. Okay, so that drug is particularly given to the patient who is undergoing this liver, lung or heart transplant. So this tacrolimus is given to suppress the immune cells of a particular recipient. Okay, up to this, any problem? Hello, any problem?
Sir, just you said that this will be tacrolimus used for liver and lung also.
Yeah, any organ transplant.
Any transplant.
Any
Okay.
organ transplant, yeah.
No, till now I feel that it's only for nephro, like only kidney transplant.
Basically that is mainly for this transplant only, but other transplant we can give this drug to just suppress the immune cells. These drugs are recommended, tacrolimus. Just note down that. Right?
Okay, sir.
Okay, thank you, Munawwar, right?
Yes. Thank you,
Munawwar.
sir.
Yeah. So this is a slide we have actually previously explained everything on this slide, clinical utility, but just to brief on that. So tacrolimus is an immunosuppressant. We have discussed this thing, as Munawwar was saying, like it is basically used for kidney transplant, but we can use it for other transplant like lung and liver also, right? So I have explained all those stuffs. The second point also, like which cell they are actually targeting to immunosuppress. T cells are basically the cells which attacks the invading or the alien molecules or cells. Okay, so we are basically suppressing these T cells. Okay, so this I think I have explained everything on this. So there are 3 different person: high responder, medium responder, and no responder, low responders, right? So we explained this. So every test will have some limitations, right? So in the case of tacrolimus, this also have some limitations, like one drug cannot treat all the all the transplant, right? As Munawar said, like are we doing this lung, liver also for this drug? Of course. So drug no, the class of drug will change, right? In that different like liver will have a different class of drug, lung have a different class of, and heart will have a different class of drugs, of course. So one test cannot define the drug level or drug type, right? Because we are targeting the variants, and that particular variants are specific towards a particular drugs. So this is the limitation. So we have to change the drug if we are changing the transplant or type of transplant organ. Okay. Yeah. So of course, correct. You are saying. So we will give you details of all those. No, which doctors you have to target for this particular test. So we'll see in the next slide. So the workflow is very simple, as I explained in the previous slide. I mean previous sessions also in NIPT, where The workflow is similar only. So blood collection will be there. Okay, processing, analysis, and reporting will be there. So this is very simple workflow for this for any kind of test. In this case, tacrolimus or pharmacogenomics. So I think this workflow don't have no problem in no problem to anyone. Do this workflow have any problem? Please go through this workflow. So sample collection will be there. I will explain you what type of tube and all. So sample collection is basically done on the EDTA tubes. Okay, 5 to 10 ml of blood is enough. Okay, so once it is come inside the lab, we will start processing. I mean DNA isolation, because we have to target the DNA or a particular variant in that, right? Then analysis will be done by lab person, and reporting will be, of course, by the lab person designated towards particular test. So as I explained, so sample collection, so collection should be like 3 to 10 ml, 5 to 10 ml EDTA blood. Okay, this blood should come within lab within 48 hours. Okay, 3 days okay, but 48 hours is our limit, because once blood is collected in a tube, coagulation starts. So to avoid those degradation of DNA and all, so 48 hours we have taken the limit. 3 days is okay, 72 hours is fine. So how we are rejecting those samples? Once samples are getting collected in a EDTA tube, it won't show any rejection kind of thing. But after some time, it start hemolyzing. Sometimes, because due to collection type or environment, temperature and all, sometimes it start hemolyzing. So we will not take those kind of sample. We immediately says that recollect those samples. Okay, so the TAT will be maximum 10 days from sample collection. I mean, from sample reception to the report generation. It won't take more than 10 days.
So the patient should be on medication while collecting this sample, or should it stop the medication before that taking sample like 24 hours?
No. Nothing like that. Nothing like that.
Okay.
Okay. Patient
Sir.
patient can be on any medications. It won't affect the test, because we are not testing the biochemistry kind of thing. We are basically testing the DNA, right? Then DNA won't change on the basis of drugs and all.
Sir, after how many? After
I'm sorry.
somebody's asking question. Okay, let them.
Yes. Yes. See, as I explained in the previous slide, right? So every individual will have a different genetic makeup. Okay. Of course, all the human beings will have a 99.9% similar this genetic makeup, but there will be a variation at particular point, or you can this is called hotspots also, you can say, or a particular point. So one person will differ from the other person at a particular nucleotide point. I mean, at a particular nucleotide, right? So what happens is genome is actually made up of chromosomes. Chromosomes actually consist of exon and introns. Okay, so exon and introns further consist of nucleotides. So on the basis of the makeup of that particular nucleotides ATGC, we are recommending drug. Okay, whether this drug is suitable for him or the other drug is suitable for him. Also, if a particular drug is suitable for him, how much amount should be given to that particular patient? Okay, and that particular test, okay, as Sayyid sir also saying, like, this is a lifelong test, okay, we won't change the dose as well as the drug type also. Okay, once it is tested, it is established. Understand? Okay, so we'll move towards the other slide. Like up to reporting, we understand. So who
Sir,
are our targeted audience? Okay,
sir,
so
two
yeah, sorry.
questions here, sir.
Yeah.
Sorry.
Yeah,
So दो question है.
no problem.
Sir, after transplant, how many days of transplant we can suggest to doctor that these tests get to be done? And second, one of my colleague asked that uh, while taking sample, like sample लेते वक्त क्या patient on drug रहना चाहिए या तो drug रोकना चाहिए?
उससे कोई इफेक्ट नहीं पड़ता.
Sir, adding to this question, sir, suppose we are testing for like tacrolimus or the clopidogrel or warfarin. So, if the patient is not taking the drug since last 15 days and we are taking the sample, so should it hamper the results?
No. See, this is not a biochemistry test, right? So, biochemistry test means the level of particular drug in a blood, right? So, we are not measuring the level of particular drug. We are recommending drugs. Okay, we are actually recommending drug, its type and its dosage
Okay, okay.
based upon the genetic makeup. Okay, so we'll move forward to the other slide. So targeted audiences. So of course, Munavar was saying like no transplant surgeons. Of course, basically they should be like no kidney, heart, lung transplant surgeons and cardio surgeons also. Any surgeon who is involved in this organ transplant, solid organ transplant. So they are our basic customers. So there is a huge market of this particular pharmacogenomics test. So there are beside this huge market, there are competitors also. Okay, so you can see Max Lab, MedGenome, Genes2Me, all these are involved in this particular test. But this is not a big market. I mean, not big competitors. I mean, these are the competitors, but no, of course, they should. If you are targeting genomics, there should be a big market for this. But right now, only 10, 12 no companies who are doing this test. Okay, and that also that FDA approved kind of thing. So we have opportunity in this. Okay, so no, this Gene Pharma, Newbergs, and Gene Omics, RCGB, Genes2Me, MedGenome, MaxLab, they are actually our competitors. So we have to compete with them in terms of that. Of course, they are doing all those, but that we have to beat, right? Now, what is our unique selling point? So unique selling point will be like we are reducing the TAT. Actually, TAT बहुत कम है हमारा 10 days. Most of the companies are giving TAT of around 12 to 14 days also. So TAT we are again working upon TAT also. Like so, we should release the report in 7 days also. So we are targeting on that. Okay, so that will increase our USP. So it is a very accurate test. Okay, and it's also it's a very fast and accurate. So these are our USPs. Coming to the reporting part, uh, there are two different tests which I have pasted over here. Two different report types. So first one is tacrolimus. The other one is clopidogrel. So you can see. We'll explain you one by one. So this is an actually a Sanger based method, right? So how many of you are aware of Sanger technology? Hello, excuse me.
Sir, it is used to identify point mutations like single gene disorders.
Yeah, no. Technology I'm asking. Anyone aware of the Sanger?
It's a sequencing method, sir. Means
Yes. It's a
that
sequencing method. Okay, it's a sequencing method. Very nice. So from there, we used to sequence the nucleotides of a particular gene. Okay, from that particular sequence, we used to fetch the information whether it is a low metabolizer, high metabolizer, or a no metabolizer, right? Based upon the genetic makeup of a particular at a particular point called variants, right? So you can see in case of tacrolimus, we are targeting these four genes, right? CYP, again CYP, ABCD, and two variant for this. So in this, you can see the RSIDs also, right? Actually represents a particular point of a variant or a particular point of a nucleotide, RSIDs. Okay, these are RSIDs. So this particular RSID have an alternate name also. Okay, so these are the names, different names for this particular point. Okay, so these are the genotype which we are saying like we will first focus upon the alternate name, right? So you can see this particular variant CYP3A4 star 1B. Okay, so you can see the alternate name for that. This is G to A. Okay, so G is a wild type, A is a mutant type or altered type. Okay, so here we are writing genotype like AA. So homozygous, right? Because AA are the same. So we are writing homozygous. Understand? So if GG will be there, we will be writing GG and wild type. Okay. Understand? So based upon this genetic makeup of these four variants, we are actually assuring and we are giving a particular drug and its doses, right? So in you can see first this tacrolimus, the other if we'll move towards this is a clopidogrel. So clopidogrel we are targeting three different variant for this, right? This is a representation of our test report. Okay, so you can say interpretation. This is a dummy report which shows like no in the view of above genotype, the patient is observed with homozygous mutant for genotype this, okay, normal genotype for this, and normal for this, and everything we are describing here. Now the last point we will say like the patient is an extensive metabolizer, star one we are giving, right? Means extensive metabolizer in the case where we have to they are the metabolizer which metabolize drugs extensively, means high amount of dose is recommended for them, right? Understand? They are the high metabolizer for of a particular drug, so we are recommending high dose for them. Understand?
Sir, means
So
you are saying that the particular drug will have less response to the patient who will be coming with more stars.
no. Star actually no. Star says it's no. Star says actually it's no. Whether it is an extensive metabolizer, is a intermediate metabolizer, or a low metabolizer. That actually is an algorithm which we have designed. Okay, based upon the study. So based upon the study and algorithm which we have designed, we are saying like it is an extensive metabolizer, low or intermediate metabolizer. Understand?
Understood, sir.
Mm-hmm. No. Don't confuse like that. See, since I explained in the previous slide, like it's a one-time test in an entire life of a particular patient, right? First point. This test is a genetic test. It's not a biochemical or biochemistry test, where uh, there will be someday there will be increased amount of something in the blood, right? Compound in the blood, low some days, but we are deciding drug, its type and dosage on the basis of their genetic makeup, which will never change. Okay, this, if this particular patient, in the case of tacrolimus, if we are saying this patient is an extensive metabolizer, so high dose of tacrolimus is recommended. So doctor will recommend this particular drug in a high concentration, or we can say high milligram, based upon its BMI. Okay, because we are, that particular patient is an extensive metabolizer, right? So 8 milligram per BMI, or milligram BMI, which doctor used to recommend. So he will or she will recommend the high dose for that particular, because this particular patient is an extensive metabolizer. He or she will never be an intermediate metabolizer in entire lifetime, because this is a genetic test. Understand?
Got it, sir.
Got it?
Yes.
Only
Sir, sir, do
Munawar
you
is only
remember?
Munawar only replies, no one other, because I think there are more than 16, 17 people. 17 people are there. Only Munawar is responding.
Sir,
14,
sir,
Dr. Santosh.
sir,
Yeah.
Santosh sir.
Yeah.
Santosh sir, do you remember this Medicover case?
Yes.
Sir, why she requested to change the reporting type, like pattern? What is the difference in our reporting and what her requirement, sir?
See, actually, no, they are actually studying this. Those doctors are actually studying, the population genetics, right? They are actually studying population genetics of a particular drug type and its dosage, right? So actually, they need this particular variant, AATT, no, something like that. They need this, and they are confused with the zygosity, like we were giving homozygous. So in that report, we have given the wild type. So they actually scared, like, what is wild type? So we have removed those wild type kind of thing, word or terminology from that report. So they are scared of that. So now it is fine.
And even the same query from me also. Sir, what is wild type, what is homozygous, and what is heterozygous?
So wild type means that particular, if a particular variant is in this case, take an example, CYP3A4 star 1B, right? So if a particular genotype G2A, okay, G is a wild type. Wild type means which is naturally present. I mean, they are not the mutant. Okay, they are not the mutant. A is a mutant. Okay, understand? So G will be
Sir,
a wild type. If genotype is GG, so we could have told them like it is a GG, and again wild type homozygous. Okay, so we'll use homozygous. We won't be using wild type because they will confuse. We will use the term homozygous again if GG will be there, double G.
okay, what is heterozygous, sir?
Heterozygosity means presence of two different nucleotide. Take an example of this, CYP last one, CYP in clopidogrel, CYP 2C19 dot star 17. So you can see CT. So there are two different nucleotides, C and T. This is heterozygosity.
Okay. Okay,
Okay.
sir.
Yeah, understand.
Sir,
Anyone?
sir, is this necessary? Like heterozygosity will have a mutant, or homozygosity will have a wild type, or
No,
it
no, no,
might
no,
be
no,
vice
no.
versa?
See, take an example of star 1B of tacrolimus, right? Star 1B. Everyone can see this slide, right? So, alternate name. First, you can see that alternate name G A, right? This science shows that G may convert into A at a particular position. Okay, the position is RSID. So, if G is there, it is called wild type. If A is there, this is called mutant type, right? So if a particular person have GG at that particular point, that particular point is RSID. If GG will be there, this is called homozygous, okay, and this is a wild type homozygous. If AA is there, in this case AA is there, again this is a mutant type homozygous, right?
Understood, sir.
Okay, fine.
Sir, is it recommended by FDA to do this test?
Yeah. FDA approved. These are the FDA approved. I'm sorry.
Sir, can you
Hello.
please share the guidelines of FDA approved recommendations?
Yes. Sure. Who is that? I'm sorry.
Sir, Deven sir, because it is very helpful for the doctors to convince them because it is FDA approved. So it is like a one step ahead to convert the doctor because it is FDA approved. They have to prescribe it.
So convincing a doctor, we are working. Actually, we had worked upon one no document which every individual will have, right? So that is actually we are designing, and actually it has been designed already. So you just have to give that particular one-page document to a doctor. Like you can read those document. If he or she not understand that particular thing, so they can come back to us, right? So we are making that document, don't worry for that. We do have document.
Dear sir, as I know, it's a doctor very well known about that. This test is approved by FDA.
Yeah.
Sometimes doctor may ask that whether you are NABL
Using.
accredited or not. But doctor, most of the transplant surgeons never ask that this test is particularly FDA approved or not because they very well known about this.
Yes, of course.
They,
You are,
dear colleague, somebody asked question, no?
yeah.
They
Yes, yes.
well known about this. Yeah, they well
Yeah.
known about this test because उनका second part यही होता है. जैसे once transplant done, कि कौन सा टेस्ट करवाना है, what drugs to be added that patient, and whether the drug is FDA approved or not, they well known about that. So no surgeon will ask this question. हाँ, might be they can ask this question that whether your lab and this year in your lab this test is NABL accredited or not. ये question वो आपको पूछेगा, but ये FDA approved है या नहीं है नहीं पूछेगा because
Yeah, of course, correct,
उनको वो जानते
100%.
हैं.
Agree.
Okay.
So this is the end of our slide, right? So and end of this training session also. So this is the point of contacts. You can write down the numbers. I think you must have this number and email IDs with you, right? So I need to understand from all the participants today, like 18 participants are there. Only three were very active in asking question. I request everyone to involve deeply on in all those slides and all those studies to ask question, and all you are open to ask the questions, please.
मयंक जी, ये अपनी स्लाइड ये रिकॉर्ड हो रही है सर? हेलो
मैम जी एक्टिव नहीं है अभी.
ओके, अच्छा ओके. क्योंकि मतलब ये थोड़ा ना बहुत ही डीप नॉलेज रेगुलर से हटके सेशन है, इसलिए मैं पूछ रहा था कि अगर कुछ ये रिकॉर्डेड होता या इसका कुछ स्लाइड्स मिलते हैं तो हमको और हमारे कलीग्स को ना इट वुड बी इजी टू रिमेम्बर.
या ओके. सो मैं अभी ना एक्चुअली एक डॉक्यूमेंट पे वर्कआउट कर रहा हूँ और डॉक्यूमेंट इज ऑलमोस्ट कंप्लीटेड, 80-90% कंप्लीटेड. सो फर्स्ट मैं ये कोशिश करूंगा, विल ट्राई टू गिव the one page document to everyone, okay, just to go through those document first, okay. So try to understand that document, and once you understand all those document, that one page document, so you can share that document to the visiting doctors. Like whenever you're visiting doctor, you can just present that particular document, color printout, one page printout. You can just give to them and ask them to read and understand those. I mean, they know it, but still that will give them confidence, right?
सर पेज ऐसा दीजिए कि हमको इजी समझ में आए और हम डॉक्टर को भी मतलब प्रमोट कर सके. जब हम ही नहीं समझेंगे तो प्रमोट नहीं कर सकते हैं. तो कुछ पंच लाइन के साथ, लाइक यूनिक यूएसपी, यूनिक सेलिंग पॉइंट्स, और ये टेस्ट हमारे पास कराने से क्या फायदा है और बाहर कराने से क्या नुकसान?
हाँ, उसमें वी आर एक्चुअली इंक्लूडिंग ऑल दोस पॉइंट व्हिच यू आर मेंशनिंग. बिसाइड दैट, पेमेंट कैसे होगा, ठीक है, कौन कैसे करेगा पेमेंट, कौन हु विल रिस्पांसिबल टू टेक द सैंपल, सैंपल कैसे आएगा,
सर
एवरीथिंग विल बी
सैंपल,
देयर.
सर रिक्वेस्ट स्मॉल, पेमेंट सैंपल कलेक्शन वगैरह तो हम सेल्स वाले देख ही लेंगे, हमको
स्टिल
सिर्फ
वी हैव
आपसे
इंक्लूडेड.
यूएसपीस चाहिए,
या
हमको
यस.
यूएसपीस चाहिए आपसे, मतलब टेस्ट के यूएसपीस दीजिए हमको, बाकी सब तो सेल्स वाले बहुत देख लेते हैं सर।
ओके, बट स्टिल वी ट्राइड, मैंने ट्राई किया है कुछ में सारे पॉइंट कवर हो जाएं, इधर उधर जाने की जरूरत नहीं, एक पेज में आपको सब मिल जाएगा. यू विल बी एबल टू हैंडल डॉक्टर वेरी इफिशिएंटली. वन पेजेस डॉक्यूमेंट, everything
थैंक यू
will be
सर,
there.
थैंक यू
Okay,
वेरी मच सर।
yeah. So shortly we will, yeah.
कांटेक्ट सुमन कलीग।
टेस्ट टेस्ट
सर जी,
का
सर जी
कॉस्ट.
प्राइस नहीं बता, साहब नहीं बता पाएंगे सर, प्राइस नहीं बता पाएंगे। यू कांटेक्ट टू सुमन इन फ्रंट डेस्क।
Yes. That's the reason I have given no contact person, right? Names and all. You can ask them. I think
फ्रंट डेस्क का हेल्पलाइन का और फ्रंट डेस्क का नंबर है आप लोगों के पास?
anyone
Who
can
raised that
contact
question?
me. Anyone can contact me. This is my contact number. Anyone can contact me anytime. I will be available to respond them. कुछ तो बोलो, आप लोग एकदम चुप हो. 18 participants हैं, but none of them are saying anything. What happened?
Sir,
Sir,
this is
clear है,
first
doubt
time where
clear
we are, listening about the pharmacogenomics. So everyone is, just I can say that they are just listening. Nobody has no experience to even represent this kind of test. So definitely, once we start in the field, we'll come up with the questions and we'll definitely come to you, sir.
Yes, sure. You people are always welcome. I'm very happy to answer all those queries whenever you come, right? So there will be a physical training also, right? So in that we will meet also and try to answer all those queries. Okay. So after this presentation, right? So this is the end of entire training program. So right now, Mayank ji will provide you the question papers, right? So अभी पता चल जाएगा कि उन्होंने कितना समझा है. लास्ट NIPT का जो हुआ था, that was very effective, and I was very impressed कि everyone understood, 70-80% they have understood NIPT. So thank you very much. Thank you all.
Thank you, sir. Thank you very much, sir.
Thank you, sir.
Thank you, sir.
Thank you for your training, sir.
Thank you very